Hello friends, welcome to the channel. Today in the lecture we will discuss about the process of sterilization and the physical agents of microbial control. Sterilization is a process that is necessarily required in the microbiology laboratories, hospitals, industries and any other place where a microbe-free or disease-free environment is required. Sterilization is the destruction or complete removal of all kind of living cells and the acellular entities from an object or habitat. Here acellular entities means organisms like the viruses which don't have a cell. Now there are various physical agents that are used for the sterilization purposes or to control the microbial population. Heat and radiations are the most commonly used physical agents. When we talk about heat sterilization can be done in two ways: dry heat sterilization and moist heat sterilization. In dry heat sterilization, incineration is the most commonly used process. In case of incineration, various incinerators or the burners are used. This is a process where the objects that are to be sterilized, they are directly exposed to the fire or flame for the sterilization purpose. This is a Bunsen burner, a kind of incinerator where direct heat is applied to the objects for the sterilization purposes. In the microbiology laboratories, the inoculation loops are sterilized using the Bunsen burners. The other method in dry heat sterilization is the use of dry oven. Now we know that what are ovens? So this is a instrument that is used for dry heat sterilization. In dry ovens, a temperature of about 160 degrees centigrade to 170 degrees centigrade is provided to the objects that are to be sterilized for a period of about 2 to 3 hours. In this duration, the microbial cells are oxidized and the cells are destroyed. This is the hot air oven where the items are sterilized at 160 degrees centigrade to 170 degrees centigrade for 2 to 3 hours. Microbial death apparently results from the oxidation of cell constituents and denaturation of proteins. The other method is the moist heat sterilization, in which steam under pressure is used for the sterilization purpose. Now autoclaves are the most commonly used apparatus that are used for moist heat sterilization. In autoclaves, steam under pressure is used for the sterilization purpose. In an autoclave, the objects are exposed to a temperature of about 121 degrees centigrade at a pressure of about 15 lb or 15 pounds for a time duration of about 15 minutes. It is believed that on exposing the objects for this time duration at this particular pressure, the microbial cells and their endospores are completely destroyed. Now both these methods, the dry heat sterilization and the moist heat sterilization, these are very effective in controlling the microbial population, but it is believed that the moist heat sterilization is a more effective process because the moist heat, that is the steam under pressure, penetrates the objects more deeply and more effectively it destroys the microbial cells. On the other hand, the dry heat sterilization, particularly the use of dry ovens, it is usually made for sterilizing the solid objects that can tolerate dry heat, like the glasswares that are used in the laboratories, talcum powder, etc. On the other hand, the moist heat is used to sterilize the media that is used in the microbiology labs or the other objects that are heat sensitive. This is how an autoclave looks like, just like a fancy pressure cooker. The development of the autoclave by Chamberlain in 1884 tremendously stimulated the growth of microbiology. In an autoclave, water is boiled to produce steam, which is released through the jacket into the autoclave's chamber. The air initially present in the chamber is forced out until the chamber is filled with saturated steam and the outlets are closed. Hot saturated steam continues to enter until the chamber reaches the desired temperature and pressure, usually 121 degrees centigrade and 15 pounds of pressure. At this temperature, saturated steam destroys all vegetative cells and endospores in a small volume of liquid within 10 to 12 minutes. Treatment is continued for at least 15 minutes to provide a margin of safety. Now, radiations are another physical agents that are widely used for the purpose of sterilization. Radiations are basically of two types: ionizing radiations, that is the X-rays, the cathode rays and the gamma rays, and non-ionizing radiations. So UV rays or the ultraviolet rays, these are the only non-ionizing radiations. The UV rays are believed to be quite lethal at a wavelength of about 260 nanometer and are widely used for the sterilization of air and the surface of the objects. The problem with UV rays is that it does not have a high penetration power. It has very low penetration power and it cannot penetrate the objects, and therefore it is used for the surface sterilization process or for the sterilization of air or water. On the other hand, the ionizing radiations, they have a very high penetration power and they are widely used for the purpose of sterilization. Now, gamma rays from a cobalt 60 source, they are most effectively used for the purpose of sterilization because they can widely and deeply penetrate the objects for sterilization. Now, here we can see an example of UV sterilization where UV light is being used for the sterilization of a chamber. UV radiation is used as a sterilizing agent only in a few specific situations. UV lamps are sometimes placed on the ceilings of rooms or in biological safety cabinets to sterilize the air and any exposed surfaces. Because UV radiation burns the skin and damages eyes, people working in such areas must be certain the UV lamps are off when the areas are in use. Commercial UV units are available for water treatment. Pathogens and other microorganisms are destroyed when a thin layer of water is passed under the lamps. That's all for the day today. Thank you.